Hello, I am Pranav and my PhD project focuses on understanding how complex traits evolve under constraints using evaluation of body size in monetary lizards as a model system. Now, complex traits are traits which are shared by multiple genes which are linked through developmental processes and the external environment in which the organism lives. Various ecological and evolutionary pressures shape how these genetic and developmental processes are expressed. Because of this multi-level organization, there are constraints which operate at every biological level. Yet, a large variety is observed in some of these traits as seen here. This capacity of biological systems to generate phenotypic variation under multiple constraints is what we refer to as evolvability. Now, there exist mechanisms in the nature through which evolution has navigated through to bypass these constraints. One way is through gene duplication, where an existing physiological gene duplicates and diversifies into new functions, such as venom components seen in venomous snakes without disrupting the original gene's roles. Another one can be thought of is ancient hybridization, where mating between two different species can enrich genetic variants, expanding the trait diversity, as seen in certain immune traits in modern humans. Constraints can also be relaxed ecologically, as seen through island gigantism, where reduced competition with other species has allowed increase in body size. Also, change in selection pressure, like during anthropogenic domestication of certain crops, can produce novel phenotypes. Now, why varanids for this project? Varanids show one of the most extreme body size variation among vertebrates, ranging from tiny species to giants like Komodo dragon. This size disparity has arisen multiple times across their evolutionary history. Like evolution of giants has happened multiple times, which can be seen in the phylogeny, although their basic body plan has remained largely conserved. Studies have shown that competition among sister lineages and habitat choice have contributed to these size shifts. But we still do not know what are the proximate causes, what are the genomic causes of this extreme size disparity. Together, this makes varanids a great model system for studying evolvability. Now, talking about the workflow, first, tissue samples are collected from multiple varanid species, followed by the generation of genome assemblies. From these genomes, various genomic features such as SNPs, copy number variants, other mutations, and epigenetic changes regulating body size evolution will be identified. These genomic patterns would be interpreted in different environmental contexts where actually these lizard species thrive. Lastly, comparative analysis which will allow us to distinguish mechanisms for generating size disparity that are shared across lineages from those that are lineage specific. Now, talking about the work progress, we have sampled tissues from different species according to their evolutionary history. Species with similar body size histories are grouped together, and we will focus on the representative species from each of the group that capture distinct evolutionary paths. Now, speaking about limitations, the analysis identifies genomic association with body size evolution rather than providing direct experimental validation. Also, environmental effects are inferred from publicly available datasets rather than through controlled experiments. Thank you.